So I figured I'd just go over the lab real quick to kind of make you feel more confident in what you learned during lab. I know it's a little crazy, we just do a lot in these labs, and they'll slow down as the semester progresses, so they're not getting any harder than from here, okay? But yeah, I just figured I'd go over the lab step by step and kind of explain each of the concepts so that you're prepared for your quiz and other exams. So for the beginning of lab, we started out by testing diffusion through a gas. Um, and diffusion, as we know, is the movement of molecules from a high concentration to a low concentration. And we saw that. So when I sprayed the perfume, it was initially at a very high concentration. Those of you who are closest to me could definitely tell because it smelled a lot. You could tell it was really, really potent. And so as we went to the next students, that concentration decreased. So what is happening to the speed of dispersion over time? In your experiment, your numbers were a little off just because of experimental error, and that's totally fine. That happens a ton. But what it should really be is it's going to slow down over time. And why that is because as these molecules are moving through other gas molecules, they're losing momentum and energy, and so they're slowing down. Okay, so which substance has the fastest diffusion rate and why? And that is going to be our diffusion through a gas. And that's because there's lots of space in between. Um, let's see if I can find it. Sorry, I have a ton of tabs up here. Okay, there's tons of space in between these gas molecules for the perfume molecules to move through. It's going to be harder if for say the same perfume molecules to move through water because there's not as many gaps to go. They're going to kind of bump into them, and then it's going to be a little bit more difficult, kind of like walking through mud versus walking through air. Okay, so diffusion through a liquid, that should have been the next easiest. See, because there is still some gaps between these liquid molecules, but not a ton, so that's why it's a bit less than the gas. All right, and semi-solid, so that's your agar dish. You put in the KMNO4, and you put that at the center, and you measure the difference in the speed. So that would have been this would have been the slowest, and that's because there's really not very much room to move. So your KMNO4 crystal molecules had a hard time maneuvering through this matrix here. And so that's the reason why it's so slow to diffuse. And that's why these substances each have different diffusion rates. All right. So in these boxes, you would have put something like this. So you put a concentrated drop of particles and in this box, and then you're drawing in the second box what happened to it. Over time, you saw in your petri dishes and your liquid dishes that the molecules did spread out, and so that's just showing that this is just more of a visual of showing you that diffusion is the movement of a substance from a more concentrated state to a less concentrated state. And the reason why it moves even isn't because the molecules are repelling away from each other, it's just because of random molecular motion. Molecules just move, and that's something to do with how much energy you're putting in and all sorts of things like that. So if I were to spray the molecules out, we'll have a pretty fast diffusion rate because that initial energy of me spraying is going to shoot them out, right? But if I'm just setting them on there, it's going to be kind of slow, and you saw that in your petri dishes. So it was slower. But yeah, it has to do with just random molecular motion, things moving around. And all molecules do that if they have energy. Okay. So diffusion across an artificial membrane. What we did for this activity, we took dialysis tubing, and dialysis tubing acts as a semi-permeable membrane. Semi-permeable means that it lets some things in and other things out. a permeable membrane would be it lets most things in, and a non-permeable membrane means it lets nothing in. So semi-permeable means that it lets some things in and others it keeps out. This whole point of this experiment was just to kind of play around and see what the dialysis tubing would let come through it. So we filled the bag halfway with 10% glucose solution, and you can remember from our first lab that glucose is a monomer of starch. It is a monosaccharide, meaning it's just one little particle and has no other extra molecular bonds. It's not bonded to another molecule, if that makes sense. Whereas starch is a bunch of glucose molecules bonded together. And we also put in starch as well, 10% starch solution. So we have these tiny molecules and these bigger molecules. And which one do you think is going to be more likely to come out of the semi-permeable membrane? And we actually found that out. So by putting iodine on the outside of the beaker, we were able to tell what went on here. So let's expand this. Okay, so this was your bag. You put iodine out here. You put glucose and starch in the bag. And we wanted to see what happened to the glucose and starch and the iodine. So, as we know from food colorimetry, iodine reacts with starch to make a purple, dark blue, or black color. So we know that the iodine entered the semi-permeable membrane because this area is purple. If starch left and iodine didn't, then starch would have gone out. It would have reacted with the iodine, and this part of the beaker would have been purpley blue. So we also wanted to figure out, well, what about glucose? We don't really know anything about glucose because we didn't have Benedict's solution. Iodine doesn't react with glucose. It only reacts with starch. Benedict's does react with glucose. And that's a harder test to do. So far in the Bio 130 labs, we haven't been successful at testing for glucose out here, but just because it's a really low concentration. But if we had done that test and if it was successful, then we would have found that yes, glucose did leave this. It went into this water. And if you ran the Benedict's test, it would have turned like a green to orange color. Okay, so from this test, we know that this membrane is semi-permeable. It lets in really small things like iodine. Iodine is really small, and glucose is really small too. So it lets glucose, small molecules like glucose and iodine pass through, while making big molecules not pass through. So the starch, the large molecule, could not pass through. And something else good to know about membranes is that nonpolar molecules and small molecules are likely to be able to pass through membranes. So I'll kind of go into that in a little bit. But that is something good to know. Okay, so keep on moving on. Alrighty. So diffusion in animal cells. This was just to kind of get you talking about hypotonic and hypertonicity. Alright, let's go back to this plasma membrane because I forgot that's the last thing we need to talk about. Okay. So, alright, here is a picture of a plasma membrane. The only thing I want you guys to pay attention to, at least for this video, is the hydrophobic tails. See this hydrophobic layer? This is the phospholipid bilayer. This is the hydrophobic layer. These heads are the hydrophilic layer. Hydrophilic likes water. Hydrophobic does not like water. In a cell there is water, and outside a cell there is water. And so that is why the heads arrange themselves this way. They're both facing towards water. And they're hiding their little hydrophobic tails on the inside so that they don't touch water because they don't like it. So one thing to know about this is that this membrane is constantly moving. These little guys are weaving in and out of each other just because of random molecular motion, and as well as these proteins. And I think that your homework mentions some of that. So we want to talk about what can go through and what can't go through. So since this layer is hydrophobic, it's a pretty big portion of the layer, if a non-polar molecule comes up to this layer and wants to get in, it typically can go through because it's attracted to this non-polar site. And if it's small enough to go between these hydrophilic regions, then it can make it through. So things that can go through are small particles that can kind of squeeze past this. Even small charged molecules can make it past, like water, for example. Water is very small, and so it can be attracted to this hydrophobic region and then squeeze through these tails and come into the membrane. Oxygen is another example of a small molecule that can kind of squeeze through, and so is carbon dioxide. Usually gases can go right between these and water, so keep those in mind. So small, uncharged, or non-polar molecules can make it through a membrane. And I think Dr. Jensen talks about that more in lecture. If she doesn't, please make sure you remember that. Okay. So let's see what else we're moving on to. All right, so we're going to talk about hypertonicity, hypotonicity, and isotonicity. We're talking about tonicity, but we're talking about hypertonic, hypotonic, and isotonic solutions. So, something I didn't mention during lab was that blood actually has this same concentration of salt on the inside of it. So, that would make you putting this salt solution outside on top of the blood. What kind of solution would that be? Well, it would be an isotonic solution. And I'll show you that in my PowerPoint if you want to look back at it. Okay. Oops, that is organic chemistry. I'm sorry, you guys. Okay, so going back to this, and we will present. Okay, so imagine you have 0.9 out here and 0.9 in here, and that's the concentration of salt. So if you have equal concentrations, then this would be considered an isotonic solution. And the water is going to move from both ends. It's going to be totally, it's random molecular motion. The water is moving in and out and in and out. But because these are equal, the water is going to be pretty equal on both sides. And I'll go more into that. All right, so here we have a red blood cell with 0.9% of NaCl, but we have nothing in here, so just pure water. So as you lessen that concentration, which you did in your lab, you added 10 drops of water. That lessened the concentration, and so this guy is going to flood with water because water is randomly moving in and out, and it's going to move from a high concentration gradient to a lower concentration gradient. So that's a hypotonic solution. In other words, osmosis is moving from a low solute gradient to a high solute gradient. You can also think about it that way. I think of osmosis like losmosis, low to high, moving water from a low solute concentration level to a higher solute concentration level. All right, and this is an example of hypertonic solution. See here, there are more solutes in the solution. It's hyper, it has lots of exciting things going on, hyper. And in here, it's still 0.9, and so the water is going to flow out because it is moving randomly, and due to randomness, the water molecules are going to tend to come out of the cell and shrink it. And that's hypertonic solution. All right, and there's this little simulation I want to show you guys. So with all that, you should have seen this in this activity. So you would have seen that this would be an isotonic solution. This would be a what? Hypertonic solution, because 10% is a lot more than 0.9%. And if you're putting blood that has a 0.9% concentration into a solution with 10% of salt, then that's going to cause issues, and the poor little thing's going to shrivel up because all its water is going to want to go towards those solutes. All right, tube 3, you should have seen, oh wow, this is going to be a hypotonic solution. You might not have seen anything at all because your blood might have popped. Your little blood cells might have exploded because all the water rushed into the cell to make everything equally distributed. Okay, and you know what? Don't worry about all of this. If you didn't see it, that's totally fine. Just understand what I told you here. Okay, so I'm going to show you a quick little simulation on diffusion. So as we increase our salt concentration, let's make this a hypertonic solution. And so we wanted to make a hypertonic solution. So the solution needs to be hypertonic, which means we have to have lots of salt in the solution and lower salt concentration in the cell. So we'll make it really different so it's easier to tell. Okay, so this is inside the cell. It only has one salt, and this salt can't pass through the membrane. What can pass through the membrane is water, and see how these water molecules are kind of moving randomly through? So due to random motion, water molecules tend to distribute equally. Over time, if you're moving things around, over time they're going to become pretty equally distributed, and you'll note that sometimes this reads 50-50. So because the salt can't move in and out of the cell, and the water is the only thing that can move, it's going to tend to equally distribute in terms of concentration. So you can watch this for a bit and kind of see how that works. So see how it's kind of harder for these guys to leave, but as soon as they do, it's easier for these guys to leave, and so the concentration is really staying the same. Okay, so let's try another situation. Let's try having the cell be at 5, and we're going to make a hypotonic solution, which means we lower this salt concentration in the solution. Well, let's see what happens. So this is inside the cell. Here's all the salt. There's a higher salt concentration inside the cell. And here is the concentration of the cell outside the cell. And there's not a bunch of salt. And so the water, has an easier time coming out of this, coming from the right to the left, because there's not all this junk going on. So you can see that the water occasionally comes in here, but then it's eventually going to hang around. It's just doing its own thing, randomly moving, and eventually it'll go back. And you'll notice that over time, due to random molecular motion, the concentrations are going to be pretty similar. And they're going to keep going until they're isotonic. And that doesn't mean that they'll stop moving. That just means that they'll be moving so constantly, randomly, that the solution concentrations will be the exact same. All right, I'll show you what that means in isotonic. So let's make an isotonic solution. Let's have the salt concentration be the same inside the cell and outside the cell, 5 and 5. All right, so you're going to notice that these numbers stay really consistent, and that's because there's equal amounts of water on the inside, equal amounts of water on the outside, and they're just kind of flipping between them. See how they're still moving, they're still going back and forth, but they're staying more consistent because they're equal on both sides to start with. So hopefully that kind of gives you a better visual of what it means to diffuse and why things diffuse and why they move from a high concentration gradient to a low concentration gradient. Okay. Alright, and this activity was just to show you kind of what happens to Elodia in a hypotonic solution and Elodia in a saltwater solution. And because it's so fun, I am really disappointed we didn't get to it. And you know, sometimes the labs just go that way and we can't get to everything, but that's okay. I'll teach you guys that I'll just show you what it's like here. So here is a video They are putting Elodea cells in a hypertonic environment. So what does that mean? That means that there's lots of solutes outside of the cell, and so where's the water going to go? The water is going to go outside the cell. So the water exited the cell. You see how they shrunk? This is their cell wall. This is the membrane. The membrane kind of shrunk away from the cell wall. Now let's add distilled water to make it a hypotonic solution. Remember, water is going to rush into the cell. You see them grow? So that's what we would have seen in lab if we did it correctly and we had time. So, yep, that's basically lab. These other two activities were just for fun. I think we all got the potato wedge thing covered. The reason why they... Oops. Oh my goodness, I'm sorry. Okay, maybe a sister's great. All right. Okay, so the potato wedges, you put it in the salt solution and it would have been really dried out, right? If you put it in the... No. If you put it in the salt solution, the salt is a hypertonic solution, and so your water would have left the potato and went towards the salt in the salt water. Now, if you put it in a freshwater solution, since there's lots of solutes in the potato, the water from the fresh water would have gone from that high concentration, or from that, it would have gone towards the high concentration of solute in the potato. And let me just visualize that real quick, because I'm doing a bad job explaining that. So, here's our potato. Let's say it has a 3 salt concentration. In the salt water, it would have a high salt concentration, right? We start, water's going to go from inside to the cell to outside the cell. You see that? And so that's why your potato kind of seemed more rigid. Versus if we put it in a really hypotonic solution, water is going to head inside the cell to make a more even distribution of water molecules. Alright, so that ends today's video. Please go to the study guide. I'm going to send that out. If you go to BYU Instructor, you go to Canvas, you will find it in your inbox. And I'm going to send that out with this video. So just go to the inbox right where this video is and click on there and you'll be good to go. Alright, thank you so much guys. Thanks for paying attention. I'm sorry lab went crazy. I hope this really helps and thanks for your patience.